An alternative approach is to inject antibody drugs through subcutaneous injection.
Okay.
But when you want to develop subcutaneous injection, the volume is very limited, right? 2 milliliter at most. So you need very high concentration of antibody. But at high concentration, some antibodies have high viscosity issue,
Right.
but it's very difficult to identify which antibody would have this high viscosity issue at early stage.
Right.
So if we can predict this behavior using sequence and structure information,
Sure.
at very early stage, it will facilitate the development of subcutaneous injection. In real world, we need to do formulation design, meaning we need to add different excipients, change the buffer, change the pH conditions. All these can affect their stability.
Right. Right.
And also the pharmaceutical companies, they are moving forward to different modalities. Okay, so I think in the next 5 or 10 years.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello and welcome to BioTalk Unzipped, where we unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin from Kansas City. And today, again, we're here in Boston at AAPS NBC. Today, we have with us in this session, we have Pin-Kuang Lai from Stevens Institute of Technology. Thanks very much for being here.
Thank you for having me.
Great, it's great to meet you. You've had some amazing strides in deep viscosity, right? And some other ML tools for predicting antibody behavior. Explain to us just briefly what deep viscosity is and what sparked your interest in combining AI with molecular engineering.
Sure. So deep viscosity is a machine learning tool that can predict high concentration viscosity of monoclonal antibody.
Wow.
So why viscosity is important for monoclonal antibody?
Yeah.
Because traditionally antibodies are administered through intravenous injection. However, intravenous injection, you need trained nurses and doctors in a hospital.
Sure.
And it takes several hours for patients. It's not very convenient.
Wow.
An alternative approach is to inject antibody drugs through subcutaneous injection.
Okay.
But when you want to develop subcutaneous injection, the volume is very limited, right? 2 milliliter at most. So you need very high concentration of antibody. But at high concentration, some antibodies have high viscosity issue.
Right.
But it's very difficult to identify which antibody would have this high viscosity issue at early stage.
Right.
So if we can predict this behavior using sequence and structure information,
Sure.
at very early stage, it will facilitate the development of subcutaneous injection.
Yeah. Well, I imagine that would save time, would save money, and maybe some discomfort for the patients, these volunteers who are going through this process. So that's great technology. Your keynote here at NBC dives deep into predicting that viscosity and bioavailability. What makes that high concentration so difficult to tackle, like traditionally?
Yeah. So as I said, viscosity is a key issue for high concentration formulation and also bioavailability, because we inject these drugs into the subcutaneous space. Eventually, we want these to go through the circulation system.
Sure.
But different antibodies, they have different permeation, like rate to the circulation system. And all these are the different factors that affect the potency of these antibody drugs. Yeah, especially at high concentration, you don't have a lot of material to test in the beginning.
Sure.
Yeah, so these are all the obstacles in developing high concentration formulation.
Excellent. Now, you've trained models in over 200 antibodies. Were there some surprising insights that kind of emerged when the data finally started telling its story? And what were those?
Yeah. So the way we develop the model is we try to combine physical features these features are like input for machine learning model. We try to combine physical features that are highly interpretable. So after we train the model, we are able to learn some physical insight into this data set. We found a lot of these data, like a lot of negative charge on the antibodies correlate with high viscosity
Sure.
and also some hydrophobicity. But we also found in some regions they show the opposite pattern, especially on the CDH3, which has the highest sequence diversity among different antibody regions. We found this quite interesting, and we're trying to delve into what's interesting, like into this behavior. Why AI tells us this pattern? Can this guide us for antibody selection and optimization?
So for the AI enthusiasts and maybe the AI skeptics out there, right, who kind of see things through the lens of like Chat GPT, how can you assure those folks that your AI results or predictions are not delusional, as Chat GPT sometimes get when you're dealing with data?
Yeah, so for machine learning, modeling, there are different validation methods. So definitely you need to test your models on a new, like independent data set that this model did not see. Yeah, there are several ways to validate the model, and we've done several validation tests and independent tests to ensure our model does not overfit,
Right.
memorize the training data.
And just so for our audience understands, this is a closed system. This isn't out on the, the internet, right? This is a system that you modeled and you built, right?
So this is the system. Yes, this is the new model.
Right,
Yeah.
right. It's not like somebody could access it, from somewhere on the internet. This is your system that you've trained specifically for these targets.
Yes, we trained this model, but we recently published the paper, and we have publicly deployed this model. So it's public.
Oh, it is. Oh,
Yeah,
so I could access it.
yeah,
And
everyone can access it.
oh, that's interesting. Okay, I did not know that. Now, you worked across Taiwan, Japan, MIT, and now Stevens. How has your international journey shaped the way you approach, scientific problems and collaboration?
Yeah, I started back in Taiwan. I started to learn like how to use like molecular modeling for different systems from like small like organic molecules.
Sure.
And then when I moved to United States, and even when I did a short visiting like research in Japan, and eventually come to United States for PhD study, I was fascinated by all the protein movement interactions and how I can use molecular modeling to study these interactions. So I started when I was in University of Minnesota for my PhD training. I started to model antimicrobial peptides, a promising alternative to traditional antibiotics. Yeah. And after I graduate, I moved to MIT, and I started to work on an antibody drug developability, assessment, and prediction. And I also started to work with a lot of pharmaceutical companies. I think when I moved from Taiwan, Japan to the United States, I see more and more industrial, like academic and industrial collaborations.
Good.
And I enjoy this transition.
Good. That's encouraging. Nice. Very good. If we fast forward 5 or 10 years from now, do you envision the future of formulation development, or how do you envision that formulation development and predictive bioengineering? And what do you think might be the boldest thing we could see that might develop in the future?
Yeah. So although we see some breakthrough in AI prediction for like monoclonal antibodies,
Right.
but now it's kind of limited to the same buffer condition. In real world, we need to do formulation design, meaning we need to add different excipients, change the buffer, change the pH conditions. All these can affect their stability.
Right. Right.
And also the pharmaceutical companies, they are moving forward to different modalities. Okay. So I think in the next 5 or 10 years, if we have a curated data set for new modalities in different formulations, then we can use AI for formulation design for novel modality. Yeah, I think this is and because we don't have a lot of data at this point for these new modalities, but I believe in the next 5 to 10 years, we can incorporate newly generated data with AI, like machine learning modeling for design. Very novel, first class modality, and with very good developability, and it can be moved forward to clinical trial and more.
Great. And you think AI is going to help speed up that process to the clinic and through the clinic?
Yes.
Nice. Awesome. Well, great. Well, my co-host, Chad, is going to ask you some questions now. Thank you.
Thank you very much.
All right.
Well, thanks for joining us today. It's good to meet you.
Yeah, good to meet you too.
Yeah. So you mentioned that this is your first AAPS meeting.
Yes.
And so I'm curious what brought you here for the meeting and what are your impressions?
Okay, yeah, I was invited by a friend. Yeah, I met a friend, and he invited me to give a talk, keynote speech on our recent publication of recent development for AI prediction of high concentration antibody developability. Okay, so I'm very honored to be invited here, and I enjoy like the format of AAPS with different. It's not like the schedule is not very busy. I have time to interact with different like attendees, speakers, and there are different like very interactive like events like in the learning lab, in these like a digital like poster presentation. Yeah, I really enjoy it so far.
Yeah, fantastic. Yeah, I really enjoyed the meeting as well. So it's good to hear. So you chose a path into academia and you're a professor now. So why did you decide to go in that direction versus going into the pharma industry and drug development directly?
Yes, yeah, a very good point. I think the academic path is, I would say, a choice of lifestyle and like my research flexibility. Because I would say in academia, I mean, both in industry and academia, you're very busy. But in academia, you're busy in a flexible way. You can decide when you want to be very busy, and when you are done with those things, you can take a few days of break. And for research flexibility, like I'm a PI, I can decide which direction I want to pursue. We can change the direction, very fast. Yeah, that's the compared to like large pharmaceutical companies,
Yeah.
you lack a lot of flexibility in choosing a research direction.
But you've had a lot of collaboration. I think the nature of your research, as you described it with Gregory, this makes sense. But you've had a lot of collaboration with pharma companies, right? I believe, right?
Mhm. Yes.
Yeah. So can you tell me, kind of the nature of some of that collaboration and how do those relationships come to be?
Okay. So I started to have collaboration with industry since I was a postdoc at MIT. Back then I collaborated with Sanofi, Merck, and AstraZeneca. I became a PI at Stevens. Yeah, I continue this collaboration with AstraZeneca. Yeah, they're also interested in developing like in silico machine learning models that can predict high concentration antibody stability. They do experiment. They measure like antibody properties at AstraZeneca. They provide us the sequence and the experimental data. We use that data and combining with our molecular modeling approach, AI approach, we develop the model and then test this model using their data set. And also, we have been collaborating in the past 4 to 5 years. And each summer, I would send 1 or 2 students to AstraZeneca for summer interns. So it's been a very like solid collaboration with them.
Yeah, it's a fantastic opportunity. Yeah. So do your models, of course, in high concentration proteins, aggregation can become a challenge. Do your models incorporate that, or they, where there's the interactions, or is it more the just the density of the molecules?
Yeah, good question. So we have a few publications before, like we predict the aggregation rate of high concentration antibodies. We have 2 publications on that. Okay. And we're also trying to expand the data set, similar to the viscosity case. We want to expand this data set for aggregation, like stability assessment. And we're aiming to develop another model for aggregation prediction in the next maybe 2 years.
Okay. Okay. So we have a number of students that watch the podcast, and if there was a student out there who was like, oh man, Dr. Lai's research sounds really interesting. What kind of students are you looking for to join your research group? What sort of skills?
Yeah, they have to. Yeah, I like to work with students that are enthusiastic about science. They're proactive. They are willing to do self learning, because for AI there are a lot of plenty of resources out there. So if they're interested, I can give them some resources. I have my own video recording for AI application on pharmaceutical development. So I've shared these resources to many students that are interested in my research area. So if they're enthusiastic about doing science, engineering, they're welcome to send me an email, and we set up a Zoom meeting to discuss a potential research project that we can work on together.
Yeah. Oh, that's fantastic. So a lot of times in research, there's other applications of your I'll call it your basic research with the machine learning models and things like that, right? You're focusing it on the this protein viscosity problem. Are there other applications that it can be applied to that are outside of that space?
Yes. So currently we are also trying to expand our approach from antibodies to other type of molecules like peptides or protein kinase. Yeah, because the strength of our lab is we're able to combine computational chemistry with data science. That's the strength of our lab. So we're trying to leverage these techniques that apply these from antibodies, different type of peptides and proteins.
Fantastic. Thank you so much. I don't have any other questions for you. It's great to meet you and to learn a little bit about your research. It's an area that I really can't say as I ever thought much about, but it's an area, it changed my perspective, right? I mean, on this area. So yeah, I really appreciate you sharing that with us. And again, it's one of the great things of the AAPS meeting is we learn things like that.
Yeah, exactly.
Yeah. And it's frankly, it's one of the reasons I love doing this podcast, because I learned these new things that I wouldn't have had these conversations otherwise.
Chad and I would like to extend a very warm thank you to AAPS for all the wonderful support, allowing us to be here and to sort of take over the media room for a while, to have some wonderful conversations with some friends and some new friends and scientists here at AAPS NBC. A special thank you to Rebecca Stouffer, who has been phenomenal in her support and helping all of this happen very smoothly. So we want to thank her, thank the organization, and we look forward to seeing you soon.